This is our DNA project. I'm Prague.
I'm Arnav.
I'm Amish, and our cameraman is Ashwat.
Alright. So first off, the keys. So this is key number 1. As you can see right over here, if you see 3 paper clips, that indicates a triple bond. You can see a blue post-it note that indicates guanine. You can see a yellow post-it note that's cytosine. Red, pink is adenine, and purple is thymine. If there's only 2 paper clips, that indicates a double bond. The green post-it note represents your cell, which is only used in RNA. The red pom-poms, or the red paper, or the pipe cleaners, represent the sugars, and the pink pom-poms, or the pink post-it notes, represent the phosphates, shown exactly like this. Alright, now we move on to key number 2. So, this is our leading strand, and for RNA, and then this is our ribose, and this is our DNA for the leading strand, and then for the lagging strand, the yellow pipe cleaner is our RNA, the purple is the DNA. So, now into key 3. This is our helicase, this is our primase, this is our primer, this is our RNase H, this is our DNA polymerase, and this is our ligase. So now into the DNA.
So, with the DNA, as you can see, we have our strands on our anti-parallel strands. This is our leading strand as our DNA is opening in this way. So this is our leading strand, and this is our lagging strand. As you can see, we have the triple bonds with the guanine and cytosine, and we have the double bonds with the adenine and thymine. And obviously, there's no uracil because that comes in RNA.
Before it can even build, we use an enzyme called primase. It lays down primer. So what primer is RNA, since the DNA polymerase cannot start from nothing, or it can start from RNA. So using the RNA primer to help to start, but when using primer, thymine gets replaced with uracil.
And this is the first part of the primer starts placing the first set of bonds or bases, and then from there the DNA polymerase takes on with just building the rest of it.
Yeah, something since this was this way, the opposite. Something true for the lagging strand. You like how lagging? It's because it's built in the opposite direction. Basically, it's built like this. So therefore, so let's say this is the first primer, or like somewhere. It builds a primer, and then it builds out, so it will fall off, meaning it will have to restart, making 2 fragments, this being the separation between the 2 fragments.
So meaning that not only does initially our primer have to start starting off the process, it also has to now start every single part where the primer starts falling off, like in this scenario, which is why you can clearly see uracil. It'll start having to put it there, and then the DNA polymerase takes on, and that happens continuously.
So now once DNA replication is done, you basically turn one strand DNA into 2 strands. However, there's still RNA. If you kill the RNA there, it'll be a mutation. That would be bad.
So which is
why
this
we take a special enzyme called RNase H,
which is responsible for taking off the uracil, which is not supposed to be in DNA,
and also the RNA.
Yeah.
However, that'll leave literal holes in the RNA. So another enzyme called ligase comes in and fills in those holes.
Yeah.
This only works at a lagging strand, however, because this one you cut off the end.
Although there will be, yes, the end will be cut off, so you won't have to worry about.
However, it doesn't really matter since the end will just be cut off permanently. So the holes in this end will be fixed, and this will just be shorter. But because of this, a cell can only really duplicate around 20 times before it fully dies out. And that's DNA replication in a nutshell.
So this is the final product of DNA replication. As you can see, we have both the leading and the lagging strands separated and attached. The uracil has been replaced with thymine, and obviously the rest of the things that have happened, like such as the primer. The primer creating a place to start for the DNA polymerase, and the same thing happened here. And essentially, this is what DNA replication.